Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today I will discuss a very important topic with you people, and it's a very unique topic. And the name of the topic is amniocentesis. Okay, and I will discuss that what really amniocentesis is, what is the procedure, why we perform this procedure, and what are the risks of this procedure, and any further details that are related to this technique. So I will discuss each and everything with you people, and after watching it I will make sure that you have get it my about the idea about the amniocentesis. Okay, so let's start with the video lecture. First of all, if I want to tell you that it is related to females amniocentesis, and it is this relates to the embryology branch of the biology. Okay, and what is the definition of the amniocentesis? So it is a prenatal diagnostic test in which a small amount of amniotic fluid is removed to determine any genetic abnormality. As we know that when the baby is present inside the womb of a mother, okay, so there is fluid that is known as amniotic fluid, and this child is present in that amniotic fluid. Okay, and we know that the child also sheds its skin cells or other type of cells in that amniotic fluid. So this amniotic fluid contains the cells of the skin cell and the other cells of the baby. Okay, so this is a type of diagnostics test when the baby is present inside the womb. So we extract that amniotic fluid from that womb by injecting a needle inside it, and after that we examine that amniotic fluid. Why we examine it and what we actually determine from it. So I will discuss it. Okay, but just keep this thing in mind that it is a medical procedure and it is a prenatal diagnostic. Prenatal means that when the child is not born. Okay, in which a small amount of amniotic fluid is removed to determine any genetic abnormality. What are the genetic abnormalities? So I will discuss it. Okay, if it is another definition, amniocentesis, also referred to as amniotic fluid test, because we are extracting the amniotic fluid from the womb, and it is a medical procedure used primarily in prenatal diagnosis of chromosomal abnormalities. Okay, what we determine the chromosomal abnormalities. The other thing is this is the chromosomal abnormality, and this is the fetal infection. We determine different fetal infections, and sex determination. We can determine also sex, that either the baby is male or female. Okay, and many other things can be determined from this test, and I will now tell you in a minute. Okay, first of all I want to tell you that it sorry that this was introduced. This technique was introduced by Robert Leslie Gad, developed the technique of amniocentesis. The accuracy of amniocentesis is about 99.4%. Okay, if we check the fluid, so and we get the result, so we can easily say that this result is 99% accurate. Okay, so it is very much fruitful technique, and the results are very accurate and precise. Okay, first of all I want to show you an overview that what really an amniocentesis is and what is the procedure. After that I will discuss each and every step in detail. Okay, this is a womb of a mother, and the baby is present, and this is the amniotic fluid. Okay, this is the amniotic fluid. What happens, we inject a needle, a special type of needle inside the woman, and we check the we check the place and the area, the exact area that where this baby is present, okay, and where should we get the amniotic fluid from. And after that we take the amniotic fluid, and we know that amniotic fluid contains cells of the baby. After that we centrifuge it, and after that what we do, we culture those cells, and then we perform different experiments over that cells. We check the chromosomes, the karyotype, we can determine sex, okay, biochemical test we conduct, and we can also use these cells for recombinant DNA technology, okay. So different result can be obtained from it, just depending on the type of experiment and the type of diagnosis we are performing from it, okay. But this is a basic overview, and now I will explain these steps in detail. First, what happened? First, if there is a person and she wants to do the amniocentesis of a child, mostly those people do this just to determine that whether her baby is a baby girl or a baby boy, and this is a very common one. But the basic is that they used to look that either their child is a healthy child or not, either it's their genetic content is in normal position, either their chromosomes are in a proper number, okay, and the different syndromes, the Down syndrome, Kleiner-Fedder syndrome, Turner syndrome, they are looking through it, okay. And for example, if a person is continuously they are having the babies and they are in abnormal condition, so what they do, so just because of because to take care of it, and just to lower the number of their babies, that for example there are 2-3 babies and they are abnormal, so what they do after that, if they are having pregnancy, okay, so in the earlier part of the pregnancy, they determine the DNA of their baby, and if it is not in a good condition, so they can also abort their pregnancy. So they also do just because of it. Or if a person is if a baby is not in a good condition and he is having any abnormality, so chances are there that it can be corrected with different techniques, and when the baby is born, so then he will be a healthy baby, okay. So there are, this is different application of amniocentesis. I was just telling you about this. What happens first? Ultrasound is performed, okay. In the ultrasound, they look to the baby, that where the baby is present, and after that, after the ultrasound, they take out the needle, and this is the needle that is used for amniocentesis. Just look to the needle, okay. This is the syringe, and this is the injecting part, okay. So this is a bit larger one, because it has to go across the skin, after that to the uterus and to the amniotic fluid. So just because of it, this is a very bigger needle. After that, what happens? They inject it inside the woman, and they are continuously performing the ultrasound just to make sure that the baby should not come in contact with the needle, okay. It is very much harmful for the baby if he or she can come in contact with that needle. After that, now here comes, this is a animated diagram. This is the syringe, okay. This is the needle. It is injected in the 90-degree angle. After that, an ultrasound is also continuously performed, or just to look that the baby is in the right position and the needle is not coming in contact with it. Okay, why the needle is so much bigger, larger? Just because it has to pass through different layers, okay. The skin, the fascia, uterine wall, and amniotic cavity, and then it has to collect that sample from it. So this is the general overview. Keep this thing in mind. Here is a procedure in the text form. A 20 milliliter of amniotic fluid should be taken from the mother. Okay, so this is a text, you can read it out. This text I have explained it in the diagrams. Okay, this is the text. After that, I want to show I told you in the earlier video that it has the first is that it has to check the normal karyotype of the baby. Okay, that either it is in a perfect condition or not. So this is a normal karyotype of a normal person. Okay, 23 pairs of chromosome, and here this is the karyotype of a male because he is having a Y chromosome. Okay, so this is a normal one. Now, why we perform amniocentesis? So it is just because we determine the sex determination. Okay, so this is the male one. If we examine the karyotype and it is just like this, so we can say that it is of a male. And if it is like this, so we will say that definitely the baby is a female baby. Okay. After that, other type of diagnosis we can do, that Down syndrome. For example, if we examine the karyotype and on the 21 number chromosome, there is a trisomy. Okay, 3 chromosomes are produced. So basically, there are 2 pairs of 21 chromosome, but in Down syndrome, there are 3 chromosomes, 21. So we can detect that the baby will be experiencing Down syndrome in the future. Okay, so there are different techniques to correct this syndrome, but I'm not going to the detail of it. Just here we examine and we diagnose the main cause of the syndrome. Okay, here is the Turner syndrome. A complete X chromosome is absent, and Turner syndrome is mostly occur in the female when they are lacking the other pair X chromosome. Okay, so this is a Turner syndrome. So it can also be determined from the amniocentesis, because when the amniotic fluid contains the cells of the baby, and when these cells are the DNA is extracted from it and it is then examined, so this is a karyotype, and it can be determined from it. Just look here, there are other syndrome, Edward syndromes. Just look here, the 18 chromosome is having its duplication 3 pairs. Okay, here come the Pateu syndrome, that the 13th pair have the 3 chromosome. Rather, it should be the 2 chromosome. Just look here, but here it is the 3 chromosome. So this is also a syndrome. Klinefelter syndrome. Just look here, the person is having the sex chromosomes are XXY. So this is the Klinefelter syndrome. Sickle cell disease, we can also determine leukemia from it. We all know that our normal body, our normal red blood cells are in a round and spherical, spongy-like type. But in sickle cell, our red blood cells are not just like the normal. It is just like in a sickle shape. Sickle shape means that it is just like this. I want to show you. This is a normal RBC. Okay, and a sickle shape RBC is just like this, and this is just because of the mutation. Okay, this is just because of the mutation. And the sickle cell disease, what happened there is abnormality and a mutation on the 11th chromosome. Okay, this is the 11th chromosome. So just look here, here is a small mutation. Just look here, this is a mutation, and if I enlarge it, so here is the HBA, and it is a normal for is GCTANCG. It is a normal gene. Okay, and what happened, it produces glutamic acid. This gene produces glutamic acid. What happens, there is a point mutation in the sickle cell disease, and here the A is replaced by the T, and after that when it is transcribed, so in place of glutamic acid, valine is produced, and just because of it, the normal RBC is transformed into sickle cell. So we can also determine this from this also from amniocentesis, and then we can deal with it. And if a person don't want to have that child, so he can also abort that pregnancy. Okay, we can also detect cystic fibrosis. We all know that what is cystic fibrosis. When there is a abnormal production of sticky mucus inside the lungs and the the pancreas. Okay, so CFTR is a type of gene that is present on the chromosome 7. If it is mutated, so these protein are basically, what does these protein do? Wait a minute. Okay, so these are cystic fibrosis, transmembrane conductance regulatory genes, protein that produces these cystic fibrosis transmembrane conductance regulatory proteins. And what does these proteins do? These proteins help in the normal uh, mm, intake of the salts and water inside the cell and outside the cell. Okay, so if it is mutated, so then abnormal mucus is produces inside the cell and over the cell. So then it can cause very much difficulties in breathing and any other type of activities. Okay, it can disturb it. So if we know about these chromosome when the person is present inside the mother, so we can then with the help of gene therapy we can replace this gene and we can correct this gene. Okay, so this is also we can deal with this disease in the earlier stage. Okay, and this is just because of the amniocentesis. After that, neural tube defects, we can also detect it and we can also correct it on the neural tubes defects. An important note, amniocentesis can also be performed in the third trimester of pregnancy because the amniotic sac and the infant is completely formed. Early amniocentesis 11 to 13 week has been studied, but carries an unacceptably highly risk for fetal injury or fetal loss, okay, of about 2.5% risk. It can also be performed in the third trimester, but it can also be performed in the earlier ages, in the earlier trimester. Okay, and after when we detect a very fatal defect, so a person can also abort its her pregnancy, because in the later if the baby is born, so she cannot deal with it, so she can abort it. Okay, so this was all about the amniocentesis. There is a small risk that an amniocentesis could cause a miscarriage. Okay, but it is very much rare, 1 in 43,000. Okay, injury to the baby or the mother, infection and preterm labor. If this technique is not performed correctly, and if the ultrasound is not performed correctly, so it might happen that the needle can come in contact with the fetus. Okay, and it can injure that fetus. And if we are performing these techniques and we are using our equipments, they are not they are contaminated, so there can be an infection in the womb of the in the uterus of the woman. Okay, and even the infection can also can come in contact with the fetus. So these are certain risk of amniocentesis. Okay, so this was all about amniocentesis. You have now known to the rare procedure that how we will perform this technique, and I have told you that why we perform these techniques. Okay, the different we can determine the sex and any other type of syndromes and diseases. Okay, if you have any further questions in your mind, you can ask me. Okay, in the comment box, I will answer you there. Okay, thank you very much. Like this video and subscribe to my channel for more interesting videos. Okay, thank you very much. God bless you.